Now let me introduce some more terms related to genetics. One term is character and the other is trait. What is a character? Character is the morphological and physiological feature of an organism. That means it is the structural feature and the functional feature of an organism. So character is the morphological, that is structural, and physiological, that is functional. Physiological feature of an organism. For example, height is a character. Color is a character. We have just mentioned height as one character and color as one character. We have not yet distinguished whether the person is tall or dwarf. We haven't distinguished whether the person is black or white. If we distinguish height as a tall and short, then it is a trait, it is not a character. If we distinguish color as white or black, then it is a trait. So, what is a trait? A trait is the distinguishing feature of a character. The distinguishing feature of a character. The distinguishing feature of a character. Suppose I choose height as the character. I choose height as the character. The person may be tall. He may be tall. And the person may be dwarf. If I take height as a character, the distinguishing feature of a character is a trait. So if height is the character, the distinguishing feature is the person is or the plant is either tall or dwarf. So tall or dwarf is the trait. And similarly, if I take color as the character, color as the character of a human being, then what are the traits? The trait is the person may be black or white. Black or white. So stating that the person is black or white is the trait of the character. Is the trait of the character. I hope you have understood what is a character and what is a trait. I will define what is a character again. The character is a morphological and a physiological feature of an organism. That is the structural and functional feature of an organism. Whereas the trait is a distinguishing feature of a character, the feature which makes the character distinct. Which makes the character distinct. The distinguishing feature of a character is a trait. Now we are going to define. Now we are going to define what is an allele. What is an allele? An allele is an alternative form of a gene. An allele is an alternative form of a gene, or an allele represents a contrasting feature of a character. It represents a contrasting feature of a character. So, what is an allele? An allele is an alternative forms of a gene. So, allele is an alternative forms of a gene. It is the alternative forms of a gene. And an allele is also called allelomorph. Allele is also called allelomorph. It is also called allelomorph. It is an alternative form of a gene. It is also called allelomorph. The term allele or allelomorph was coined by Bergson. It was coined by Bergson. Remember, it is Bergson who coined the term genetics. He coined the term genetics. And it is Bergson who is also called the father of modern genetics. The father of genetics is Richard Johann Mendel, remember? But the father of modern genetics is Bergson. So it is Bergson who coined the term genetics, and it is Bergson who is also called the father of modern genetics, and it is Bergson who coined the term allele or allelomorph. Allele or allelomorph. So an allele represents the contrasting character, contrasting pair of a character, or an allele represents the alternative forms of a gene. And you have learned that a gene is located at a point called locus on the chromosome. Now let me explain where are these alleles located. Where are these alleles located? Suppose this is a chromosome. This is a chromosome with an arm or chromatid. Imagine that this is the chromatid of a chromosome. This is a chromosome. Of course, it has a two chromatids, and this is another chromosome with a centromere, and this is a chromatid. I will take this as a locus. Locus is the point at which the gene is located. Locus is the point at which the gene is located. And this is also a locus at which the gene is located. And these two chromosomes are morphologically similar. Therefore, these chromosomes are homologous chromosomes. Homologous chromosomes. Suppose there is a gene. Let us assume the gene is a T at this locus, and at the same locus, on homologous pair of chromosome, there is a gene T. Try to understand. Or you can take small t gene here or small t gene here also. You can take capital T gene here and small t gene here. My intention is to define what exactly an allele is. An allele is the alternative forms of a gene. Alternative means two forms of a gene. That is the meaning of alternative. So sometimes you may be given a question, what is the alternative method? Means other than this. So an allele is the alternative form of a gene located at the same locus on the homologous pair of chromosomes. That is the definition of an allele. An allele is the alternative forms of a gene located at the same locus on homologous pair of chromosomes. What are homologous chromosomes? Chromosomes which are morphologically similar. Yesterday we discussed what are homologous chromosomes and what are non-homologous chromosomes. So let me define allele. An allele is alternative forms of a gene. Alternative forms of a gene located at same loci or different locus. It is at the same locus. The positions of the genes are the same on homologous chromosomes. At the same locus or at the same locus of homologous means structurally similar. Homologous pair of chromosomes. Pair of chromosomes. So that is an allele. An allele is also called allelomorph. An allele is also called allelomorph one, and an allele is the alternative forms of a gene located at the same locus on homologous pair of chromosomes. That is an allele, and the term allele was coined by Bateson, and it is a Bateson who also coined the term genetics, and it is a Bateson who is also called the father of modern genetics. Don't get confused. If the question is father of genetics, it is Mendel. If the question is father of modern genetics, it is Bateson. If the question is the father of experimental genetics, it is Margon. If the question is the father of human genetics, then it is Garrod. Don't get confused. If I repeat, if the question is father of genetics, it is Mendel. If the question is the father of modern genetics, it is Bateson. If the question is father of experimental genetics, it is Morgan. If the question is father of human genetics, then it is Garrod. So we try to remember Morgan as the father of Experimental genetics. Father of experimental genetics is Morgan, whereas Gerard is called the father of human genetics. The father of human genetics. I hope you have understood what is an allele. Now we are going to discuss some more terms like what is homozygous and what is heterozygous condition. Now let me define what is homozygous and what is heterozygous conditions. Okay, let me define what is homozygous condition and what is heterozygous condition. What is homozygous and what is heterozygous? What is homozygous condition? And what is heterozygous condition? I already told in the introductory class itself, the command on the terminology of genetics is more important for easy understanding and delivering of the subject. Remember. So, you should have a thorough knowledge of this terminology. If you have a command over this terminology of genetics, you can easily understand the various phenomena that happen in the process of inheritance. Now, let me define first what is a homozygous condition and what is a heterozygous condition. Now, let me define first what is homozygous. Again, imagine that this is a chromosome with a centromere. This is the centromere of a chromosome. Imagine that this is a chromatid, the arm of the chromosome. Imagine that this is one chromosome and this is another chromosome. They are identical chromosomes or homologous chromosomes. Now I will take this as a locus on one chromosome and this is the locus on the other chromosome. The positions of the genes on both the chromosomes is at the same locus. Let the gene represented is a T. I will take capital T here. On the other chromosome, I will take capital T gene at the same locus. Now, what is homozygous? Homozygous means when an allele has similar genes. When an allele has a similar genes. What are similar genes here? Capital T gene on one locus and capital T gene on the locus of the other chromosome. An allele with similar genes is homozygous. So what is homozygous? Homozygous means an allele possessing similar or identical genes. An allele possessing similar or identical genes is homozygous condition. So in this particular case, TT is homozygous. Or if I say small t here and small t here, then it is also homozygous. So you can have many examples. If I say capital A for one character, then it is homozygous condition, and small a, it is also homozygous. An allele has a tail of genes, you should remember. An allele possessing means carrying identical genes. Both are capital T genes or both are small t genes. Or both are capital A genes or both are small a genes. Then the condition is said to be homozygous. So the condition is said to be homozygous when an allele has identical or similar genes. Now observe this diagram. Now this is again a chromosome with a centromere and chromatids. Imagine these are the homologous chromosomes. These are structurally similar chromosomes. At this locus, there is a gene T. At the same locus on the other chromosome, there is a gene T, small t. So at this locus, there is a capital T gene, and at this locus of this chromosome, there is a small t gene. Now, what is an allele here? An allele possessing non-identical, that is dissimilar, non-identical genes. Non-identical genes. One is a capital T gene and the other is a small t gene. Non-identical genes or dissimilar genes, then the condition is called heterozygous. I hope you have understood what is homozygous condition and what is heterozygous condition. The homozygous condition, in homozygous condition, an allele shows similar or identical genes. Similar or identical genes. In heterozygous condition, an allele shows non-identical genes or dissimilar genes. One is a capital T gene and the other is small t. Therefore, this is heterozygous. And other example is heterozygous, capital A, small t. And similarly, we can have a number of examples, capital Y, small y, heterozygous. We can have a number of examples. You can have a number of examples. So I hope you have understood what is homozygous condition and what is heterozygous condition. When an allele shows similar or identical genes, then the condition is said to be homozygous. When an allele shows non-identical genes or dissimilar genes, then the condition is said to be heterozygous condition. Examples of homozygous condition are capital T or small t. An example of heterozygous condition is capital T small t. I hope you have understood what is homozygous and what is heterozygous. Then we are going to define then what is a dominant character and what is a recessive character. What is dominant and what is recessive. Now let me explain what is dominant and what is recessive. Now what is dominant and what is recessive? Suppose for the character height, if I take capital T for tall, capital T for tall or tallness, and small t for dwarfness, I have taken capital T as a tallness and small t as dwarfness, that is short. If I represent capital T, capital T, what is the condition? The condition is homozygous. Homozygous because it has identical genes, therefore this is homozygous. Homozygous tall. Homozygous tall. I already told you I have taken capital T representing tallness and small t representing dwarfness or short character. Now I will write this in heterozygous condition. What is the heterozygous condition? One is a capital T and the other is a small t. This is heterozygous condition, therefore it is heterozygous. Heterozygous, again it is a tall. Heterozygous tall. I will explain later. Can understand in a better way? I will explain. Capital T is a homozygous tall and capital T small t is heterozygous tall. If I write small t, both are identical genes of an allele. The small t are homozygous. Homozygous dwarf. Small t represents dwarf condition. or short stature or dwarf condition. Now, with this basic concept, we are going to define now what is dominant character or dominant feature and what is a recessive feature. If you observe these three conditions, capital T, capital T, capital T, small t, small t, small t, you can easily define what is the dominance and what is the recessive. The character which is expressed in both homozygous condition and heterozygous condition is a dominant. The tallness is expressed in capital T condition, that is in homozygous condition. The tallness is expressed in heterozygous condition also, that is in capital T small t condition. Therefore, what is a dominant character? The character which is expressed in both homozygous condition and also heterozygous condition is called a dominant character. So, what is a dominant character? The dominant character, a character which is expressed, which is exhibited, which is expressed or which is exhibited in both homozygous and heterozygous condition is a dominant character. Is a dominant character. So tallness is expressed in this homozygous condition, capital T, and tallness is also expressed in heterozygous condition. So tall is a dominant over dwarf. Tall is a dominant over dwarf. So the character tallness is expressed in homozygous condition and heterozygous condition. Therefore, the character which is expressed in homozygous condition as well as heterozygous condition is called the dominant character. Can't you define what is heterozygous? What is the recessive character? Now observe this. Small t. Tallness is a dominant character, whereas dwarfness or short stature is a recessive character. The recessive character is expressed in homozygous condition only. This is homozygous condition. Though there is a recessive gene, small k gene, in heterozygous condition, though there is a recessive gene, small p gene, observe, look at this. Though there is a recessive gene, small p gene, in heterozygous condition, dwarfness is not expressed due to the dominance of tallness. Dwarfness is not expressed due to the dominance of tallness. In this case, both the genes are small t genes, identical, they are homozygous to dwarf. Therefore, what is a recessive character? The character which is expressed in homozygous condition only. In homozygous condition only. Is a recessive character. Is a recessive character. So this is a dominant character and this is recessive character. Sir, start చేసి Let's start చేసి తర్వాత స్టార్ట్ చేసి ఎంత సార్ సరే ఇంకా కంటిన్యూ చేసి సార్ మీరు నేను చూడిపోయి వాళ్ళని తర్వాత ఎడిట్ చేసుకోవచ్చు ఇది చేసేస్తున్నారు ఈ గ్యాప్ ఎడిట్ చేసుకుంటాను ఓకే Now let me explain the other two terms like what is the phenotype and what is genotype. Now we are going to discuss what is the phenotype and what is the genotype. What is the phenotype and what is genotype? Now I will consider a plant, that the plant is a pea plant. On observation, I say it is a tall plant. It is a tall plant. When I say it is a tall plant, you are not sure that whether it is homozygous stalk or heterozygous stalk. Homozygous stalk means capital T genes with capital T genes. Heterozygous stalk means with a capital T small t genes. If I on mere observation, I say it is a tall pea plant. When I say it is a tall pea plant, a tall person, we are not sure whether he is homozygous tall or heterozygous tall. We define the tallness based on the external appearance. Then what is phenotype? The phenotype is the external makeup of an organism. It is the external appearance of an organism. Some persons externally they may look very kind. The external look is very kind, but internally they are very harsh. We have not analyzed the genetic makeup of the person based on the external features. You have come to a conclusion that the person is kind. Based on the external features of the plant, you have just come to a conclusion that it is a tall plant. But we are uncertain whether it is homozygous tall or heterozygous tall. So what is phenotype? Phenotype refers to the external makeup or appearance of an organism or an individual. So what is phenotype? Phenotype refers to the external appearance or makeup of an organism or individual. Saying that tall plant, but we are not sure whether it is homozygous tall or heterozygous tall. We have not analyzed the genetic makeup of an organism. We have stated the character based on the external appearance. So, the external makeup or the external appearance of an organism or an individual is its phenotype. Then what is genotype? What is a genotype? Suppose I say it is homozygous star. When I say it is homozygous star, immediately it should come into your mind that the condition is capital T, capital T. When I say it is dwarf, dwarf is always expressed in homozygous condition. Therefore, it is a small t, small t. If I say heterozygous star, then you have to remember that it is a capital T, small t. Then what is a genotype? The genotype is the genetic makeup or the genetic constitution of an individual. The genetic makeup of an individual is its genotype. The external appearance of an individual is its phenotype. So phenotype is the external appearance of an individual, whereas genotype refers to the genetic makeup of an individual or a plant or an organism. That is genotype. So stating homozygous tall, stating heterozygous tall is expressing its genotype. Stating tall is expressing the character in phenotype. If I say tall plant, we are not sure whether it is homologous tall or heterologous tall. If I say homologous tall or heterologous tall, it is set based on the genetic makeup, the association of genes. That is capital T association make it homologous star. Capital T small t gene association make it heterologous star. I hope you have understood what is a phenotype and what is a genotype. The term phenotype refers to the external appearance of an organism or individual, whereas the term genotype refers to the genetic makeup or genetic constitution of an organism or an individual. Both the terms phenotype and genotype, both the terms phenotype and genotype, the terms phenotype and genotype are coined by Johansson. I said Johansson also coined the term gene. It is Johansson who coined the term gene. It is Johansson who also coined the terms phenotype and genotype. It is Bateson, it is a recollection. It is Bateson who coined the term genetics. It is Bateson who coined the term allele. It is Bateson who is called father of modern genetics. So this is only a recollection of what I said earlier. Now we are going to discuss a few more important terms. They are terms like karyotype, ideogram, and ploidy. Now we are going to discuss these three terms. What is a karyotype and what is uh, ideogram, and what is ploidy. Sometimes you may come across the words like cryptograms, phanidograms, cryptogram, phanidogram, cryptograms. What is or what are cryptograms? Cryptograms are non-flowering plants. Non-flowering plants like the thallophytes, bryophytes, pteridophytes. They are cryptogams. They are non-flowering plants. Then what are phanerogams? Phanerogams are flowering plants. They are also called anthophytes. Then what is a cryptogram? Don't get confused. The purpose of introducing these terms is to make you aware of a question in NEET examination. Sometimes they use this terminology and identify the mismatch pair or make a correct matching. In such case, the knowledge of these different terms is useful. I repeat, cryptogams are non-flowering plants. Phanerogams are anthophytes, are flowering plants. And what is a cryptogram? A cryptogram is the code describing a virus. The code describing a virus is cryptogram. The code describing a virus. The code describing a virus is cryptogram. Now let me define what is karyotype, what is ideogram, and what is ploidy. What is a karyotype? The karyotype describes the morphological features of a chromosome. That means the size of the chromosome, the shape of the chromosome, and the distribution of the chromosome. In brief, its structure. Structure includes the size, shape or form of the chromosome and also its distribution and also their distribution. So karyotype refers to the morphology of chromosome. Karyotype refers to the morphological features of a chromosome. It refers to the morphological features of a chromosome. It is karyotype. Suppose I say the chromosome has a centromere at the center and its chromatids are equal size. Then it is its karyotype. I said it has a chromosome at the center with the equal sized chromatids. That means I am speaking about the metacentric chromosome, which is V-shaped. So saying that the centromere is at the center of the chromosome with equal sized chromatids or arms is its karyotype. Now, I will represent this in the form of a diagram. This is the centromere of a chromosome, and these are the chromatids. Now in the second case, I try to represent the morphology of a chromosome with the help of a diagram. The diagrammatic representation of karyotype is ideogram. So this is ideogram. Ideogram of chromosome. So what is ideogram? Ideogram is the diagrammatic representation of a chromosome. That means it is the diagrammatic representation of a morphology of a chromosome. The technical description of a morphology of a chromosome is its karyotype. It is the technical description of the morphology of a chromosome. Whereas the diagrammatic representation of karyotype is ideogram. If the chromosomal structure, the distribution, their arrangement are diagrammatically represented, then it is called ideogram. So the diagrammatic representation of a chromosome is called ideogram. Then what is ploidy? And what is a ploidy? Then ploidy refers to the number of chromosomes. The number of chromosomes. Ploidy refers to the number of chromosomes. Suppose in somatic cells or body cells, they are also called body cells, or in mother cells, maybe pollen mother cells or sperm mother cells, or megaspore mother cells, microspore mother cells. Or in zygote. A zygote is the result of the fusion of male and female gametes. It is the result of fusion of male and female gametes. So I have given the example of somatic cells, mother cells, microspore mother cells, megaspore mother cells, or pollen mother cells, or sperm mother cells, etc. and zygote. In all these cases, you can see 2n set of chromosomes. 2n set of chromosomes. When a somatic cell has a 2n set of chromosomes, say for example, when a human cell has a 46 chromosomes, it is a 2n set of chromosomes. When a human cell has 46 chromosomes, it is a 2n set of chromosomes. When Drosophila, which is also called the fruit fly, has 8 chromosomes, then it is a 2n set of chromosomes. When cotton pea plant has 14 chromosomes, it is 2n set of chromosomes. I have given only a few examples. The number of chromosomes in the somatic cell or body cell or sperm mother cell or ovum mother cell or the zygote of human being, the number of chromosomes is 46. That is 2n set of chromosomes. If a cell has a 2n set of chromosomes, say for example 46 in human beings, 8 in Drosophila, 14 in pea plant, garden pea plant, then it is called diploid. So, having 2n set of chromosomes is diploid condition. Having 2n set of chromosomes is a diploid condition. Half of diploid. What is half of diploid? Half of diploid means it is 2n by 2. That is half of diploid. Half of diploid, that means having n set of chromosomes in a cell. N set of chromosomes in a cell. It is a half of diploid. It is haploid. Half of diploid. Half of diploid is haploid. So having n set of chromosomes is a haploid condition. Having 2n set of chromosomes is diploid condition. Diploid condition. And n set of chromosomes, that is a haploid condition, is seen in gametes such as male gametes like sperms and female gametes like ovum. I said human being has 46 chromosomes. Out of 46 chromosomes in human being, maybe male or female, the father has to contribute to 23 chromosomes through the sperm, and the mother has to contribute 23 chromosomes through her ovum. Therefore, the child has to get 46 chromosomes in total if there are no chromosomal abnormalities. If there are no chromosomal abnormalities. So, you must understand what is ploidy. Ploidy refers to the number of chromosomes. If the number of chromosomes is a 2n set of chromosomes per cell, then the condition is a diploid. Such a diploid condition is seen in somatic cells or body cells or mother cells or zygote. Whereas having n set of chromosomes per cell is a haploid condition. Such haploid condition is seen in gametes like sperms or microspores or pollen grains. They are also haploid. Ovum is also haploid. Ovum is also haploid. Now, having 3n set of chromosomes. Having 3n set of chromosomes. When there are 3n set of chromosomes, what is it called? It is called triploid condition. Triploid. Having 3n set of chromosomes is a triploid condition. Such a triploid nature is seen in endosperm of plants. In the endosperm of plants is a triploid in nature, especially in the endosperm of angiosperms. In angiosperms, The endosperm is the result of post-fertilization. It is the result of post-fertilization and it is a triploid in nature with a 3n set of chromosomes. Whereas in gymnosperms, the endosperm is formed before fertilization and in gymnosperms, the endosperm is haploid. Presence of haploid endosperm is a characteristic feature of gymnosperms. Remember, gymnosperms such as cycas is a gymnosperm, pinus is a gymnosperm, netum is a gymnosperm, ephedra is a gymnosperm. So there are a number of examples for gymnosperms. Now my intention is to make you aware of the ploidy, that is the number of chromosomes. There is ploidy like a haploid condition, there is a triploid condition, there is a diploid condition, and if the chromosomal number exceeds 3n, that is 4n, 5n, 6n and so on, then the condition is said to be polyploidy. If a cell has more than 3n set of chromosomes, that is 4n, 5n, 6n and so on, then the condition is called polyploidy, and such polyploid condition is more common in plants and less common or rare in animals. Remember, try to remember this point. The polyploidy condition, it is more common in plants and it is very rare or less common in animals. And this polyploid condition, what is polyploidy? Ploidy refers to number of chromosomes more than 3n, it is 4n, 5n, 6n, 7n and so on. Such polyploidy condition is very common in plants, I said, and it can be induced by treating with the chemicals. For example, there is a chemical called colchicine. The colchicine is a chemical. It is an alkaloid obtained from a liliaceae member called Colchicinum autumnale. This Colchicinum autumnale is a member of Liliaceae family, from which the alkaloid colchicine is obtained. And this colchicine and alkaloid obtained from Colchicinum autumnale, which is also called meadow saffron. Try to remember this. Meadow saffron is the common name of Colchicinum autumnale. Meadow saffron belongs to family Liliaceae, and this colchicine, it induces polyploidy in plants. Remember, when a dividing cell, say for example, when a cell dividing mitotically is treated with colchicine, when it is treated with colchicine, the cell division is arrested at the metaphase. The cell division is arrested at metaphase. When the cell division is arrested at metaphase stage, there is an increase in the number of chromosomes, leading to polyploid condition. Therefore, the chemical which is most commonly used in inducing polyploidy in plants is colchicine, and this colchicine is obtained from a plant called Colchicinum autumnale. It is commonly called meadow saffron plant. It belongs to family Liliaceae. So, to try to remember all these points also, first we try to remember what is a diploid, what is a haploid, and what is a triploid. And triploid condition, for example, it is seen in endosperm of angiosperms. The endosperm of angiosperms is triploid in nature. So this is about ploidy. Then based on the ploidy, we have to know what is aneuploidy. So far we have discussed what is euploidy. This is a N condition, haploid, 2M condition, diploid, 3n condition, triploid, 4n condition, you can say tetraploid, 5n condition is a pentaploid. All these conditions they refer to euploidy. They refer to euploidy. Now we have to understand what is aneuploidy. Unless you understand the concept of aneuploidy, you cannot understand the genic balance theory of bridges. The genic balancing of in the genic balancing of bridges, you will come across the word aneuploid condition of the dipole. If you know what is this aneuploid condition, you can understand better what is the genic balance theory, and you can understand better how the syndromes are formed due to the chromosomal abnormalities, especially in the number of chromosomes, like Klinefelter-Stratton syndrome, Turner's syndrome, and other autosomal syndromes. So the chromosomal abnormalities can be better understood if we know what is this aneuploid condition. So we are going to discuss aneuploidy also.